Good morning, science lovers. So last year, the topics that were covered were mostly physical sciences. We talked about the Big Bang, etc. And this year, we're concentrating more on life sciences and social sciences. The first half of this year's program series is about life sciences. Of all the topics last year, I believe that the way that we covered the topic that we wrestled with around symmetry theories and physics was the most difficult conceptually and in terms of the amount of content to get through. I suspect that of all the programs this year, this is probably the most complex to try and educate a curious lay audience about. Because there's a lot of special terminology and whatnot. I'll try to either define my terms as I go or avoid using terms that are overly technical. If I do use a term that you don't understand, you don't know what it means, please don't hesitate to interrupt me and ask me what the heck I'm talking about. Next time we're going to talk about the Gaia hypothesis, essentially. A little bit of a customized take on the notion from my point of view, customized, that Earth and the various physical systems of Earth, including geology and atmospherics, are implicated in a vast feedback network with life, and some of the effects that has on both domains, both natural, geology, atmospheric, oceanic, and life. So if you want to find out more about what we do as the Humanists of West Suburban Chicagoland, the best place to keep up with events and happenings, as well as providing a mechanism for a discussion group, is our meetup group. And we also have a YouTube channel where these lectures, as well as many of our other programs, are presented as videos. Last week we had our Humanist program talking about DNA and exoneration with regards to criminals or accused criminals that were found innocent because of DNA evidence. So that was a nice segue into today's topic about DNA, genetics, and epigenetics. Our Humanist regular monthly meetings occur on the second Wednesday of every month at 7:30 p.m. And again, keep up to date with what we're planning by virtue of the Humanists of West Suburban Chicagoland meetup group. I'd also like to thank this church for providing facilities, microphones, lights, a roof over our heads, etc. And like all establishments, it doesn't run on good wishes. So there is an offering bowl placed back there if anybody feels like contributing. And that's all I'll say about that. All right, DNA, genetics, and epigenetics. You know, the old story was that every cell, with a couple of exceptions like the red blood cells in animals, every cell has a DNA molecule, or several DNA molecules wrapped up in chromosomes, and that those DNA molecules encoded genes, and that those genes express proteins. Now, proteins are amazing molecular machines. They can do any number of things. You've heard about hormones, you've heard about enzymes, you've heard about things like muscle fibers, perhaps made of actin and myosin. Nearly every structure in your body is made of proteins. So a gene encoding a protein is a very fundamental aspect of biology. And for many years, for generations of scientists, it had been expected that any genome, which is the collection of information recorded in it in the DNA, any genome would have innumerable, almost, numbers of genes, and that was the way the program or the code for life was both recorded and implemented. Well, now there's a new story that DNA is full of genes to the tune for human beings of about 2%. So about 2% of our DNA codes for genes. And for the longest time, it has been considered that the rest of it is junk. We're going to talk about what that implies. Previously, in this year's lecture series, we've talked about ideas regarding how life may have plausibly arisen from natural processes, geologic and physical and chemical processes on the planet Earth, and that those processes are likely to be amenable to the climates of other planets as well. So we don't necessarily expect that life will only exist on Earth. We've talked about the concept of natural selection by virtue, or evolution by virtue of natural selection. We talked about that last time. And this time we're going to talk about the mechanism for natural selection and passing information down generations. As we discussed last time, there were some questions that stumped Darwin. One of Darwin's problems, it delights me to talk about, it's not related to genetics, but I like reiterating it, is the young Earth problem. So Lord Kelvin, who was active at the same time as Darwin, he was the preeminent physicist of the day, did some thermodynamic calculations and decided that the sun could be about 10 million years old, assuming that it was a chemical fire or that the heat of the sun was generated by virtue of gravitational collapse. And Darwin didn't have enough time. He recognized that natural selection required far more than just 10 million years. 10 million years was itself an inconceivable amount of time compared to the conventional wisdom of the day that the Earth was only several thousand years old. So Darwin was set against conventional wisdom about the age of the sun and the Earth, just by virtue of his studies of biology. Another problem that he wrestled with, of course we now know that the sun uses nuclear processes, as we talked about last year, and is billions of years old, as is the Earth. He also was struggling with the idea of missing fossils. Fossils are in the ground, often deep underground, and they're hard to find. And we talked last time about missing links and how that's associated to missing fossils and how it's kind of a misconception about the way science works and the way evolution works. This notion that every time you find a new intermediate species between existing species, you've created 2 more gaps that you need to fill, and then so on and so on, 4 gaps and 8 gaps, etc. Darwin was perplexed by the mechanism of inheritance. Clearly, parents passed on their physical being. That the fact that you have 2 arms and 2 eyes and look like a human instead of a chipmunk. These kinds of things are passed from parent to child, but he had no idea about the physical mechanism that underlie this. He did, however, have great confidence that there was, in fact, a physical mechanism, that there was nothing mystical or magical involved. He just didn't know what it was. And then similarly, he didn't know what the mechanism was for gradual change across time. What is the actual physical mechanism that facilitates gradual change? Is the agent of evolution, as it were? These were questions that he did not have the answer to. About the same time as Darwin was doing his work, a fellow by the name of Mendel was doing some work with peas. And he found that through cross-breeding different peas and breeding strains of peas for characteristics that look to be completely heritable from generation to generation, he came up with a mathematical specification of the way that inheritance works with regards to what we call Mendelian genetics. He still didn't know what the mechanism was. How did peas encode for yellow versus green seed pods, for example? But he could see that the exhibition of those characteristics across generations followed clear and quite literally mathematically discernible patterns. Then, in the 1900s, in the 20th century, scientists discovered that DNA was the basis for genetic information, inheritance, and it was also the structure amenable to mutation. So it was also the means of conveyance of heritable changes and mutation. Many of you have heard about DNA. If there is any single iconic image of science in the 20th century, it's the double helix. The notion that you have paired bases, nucleotides on the DNA that form this kind of ladder structure that twists around itself in this spiral pattern. Watson and Crick received the Nobel Prize for uncovering the structure of DNA in 1953. We've since discovered that DNA and its nucleotide bases coming in triplets encode for 21 amino acids that are expressed in nearly all living things, including human beings. Of those 21 amino acids, we have the metabolic capability to make about 11. So we need to eat things to get all the amino acids we need. The DNA in each typical human cell is encoded across 23 pairs of chromosomes. So you may remember that from some science show or biology in school, that the DNA is not one single large molecule nearly each of our cells, but rather is divided into 23 pairs of chromosomes. If you were to take all of the DNA in a single human cell and stretch it out end to end, it would make a chain about 4 feet long. And think about that. That's a molecule, or it happens to be several molecules in this case, but the molecules are literally so big, they're essentially macroscopic. Molecules that are big enough to have a practical, discernible physical extent that is not thick enough to be seen with the naked eye, but certainly long enough that we could potentially see them. The DNA, however, in chromosomes is balled up inside the cells. And the mechanism for balling up that DNA and keeping it literally tightly wound, falls to these things called histones, which form a backbone around which chromosomes wind the DNA. We also have mitochondrial DNA. So we have about 20,000 genes in the human genome, but everybody also has mitochondria in their cells, and those mitochondria encode for an astonishing 37 genes. The human DNA has about 3 billion base pairs, and the mitochondria has 16,000, just over 16,000. So why is it that we have 2 different DNAs? We talked a little bit about that in the lecture from a couple months ago regarding the origin theories for the evolution, or the origin of life on Earth. And one of the things we covered in there was the role that mitochondria play. If you weren't here for that, I recommend looking for it on YouTube. All right, so let's talk about relative DNA sizes. So we already said that humans have about 20,000 genes in their spread across their 3 billion bits of information across all the DNA. Here's something with a relatively small genome. This is an Antarctic midge. It has 99 million base pairs as opposed to 3 billion. And that number is probably small to facilitate survival in an extremely harsh environment. So its cells have to reproduce relatively quickly, right? The fewer the number of base pairs, the smaller the amount of DNA that you have to copy while going through your metabolic processes, the faster you can do it. The machinery to copy DNA and genetic information operates in finite time. So the larger the genome, the more time it takes for cell division. As I said, the human DNA is about 3 billion base pairs. Another way to look at the human DNA, there was a program I saw and I apologize, I can't remember which program, one of these science documentaries that is available. A man was walking through a library, row after row, shelf after shelf of large books that had big white bindings. They looked like a modern dictionary. They were so big. And it turns out that if you were to encode human DNA without putting any spaces in between the letters, the A, G, T, and C, that comprise the information stored in DNA, you would end up with 1,000 dictionary-sized books. A library of 1,000 books of 1,000 pages each. It's a huge amount of information. And then we have this beautiful flower, Paris japonica. It's a Japanese flower. And it has 15 billion base pairs of DNA. It's about 30 feet of DNA if you were to stretch it all out. And that's very interesting. One of the things that I said about the midge is perhaps one of the reasons its DNA is small is because it lives in a very harsh environment. Well, this particular flower lives in an accommodating environment. Plenty of rain, plenty of sunshine, not many predators. So it seems that nature finds a balancing act between putting a lot of information in a genome and making it quite literally easier to deal with from a biochemical standpoint. As I said, the human DNA has about 3 billion base pairs. This thing has 15 billion base pairs. This is our humble rice. Rice, you may be relieved to know, has about 10 times fewer base pairs than human DNA. That's the good news. The intimidating news is that it has over twice the number of genes. So what's going on here? Why is it that something as complex, we consider ourselves the pinnacle of evolution, how is it that humans, this supposed pinnacle, have 20,000 genes, but a humble rice plant has 50,000 genes? What's going on here? And furthermore, what's going on with all the junk in our DNA? Remember, I said that this rice plant has a genome that's about 10 times smaller in terms of the actual size of the DNA, 10 times smaller than human genomes, and yet it has more genes. Maybe there's something more to DNA than just coding for proteins, just genes, and we'll talk about that. Also, an interesting aside, amphibians tend to have longer genomes than other animals. Amphibians go through a very complex life cycle. They also have to have a physiology and metabolism concordant with both living on land and in water. Probably takes a fairly complex biochemical toolkit to be able to do that.
They can regenerate organs too.
In many cases, yeah. All right. So what is all this junk that's inside our DNA? Back in the 1970s and 1980s, early 1990s, as I said, it was discovered that only about 2%, it's actually about 1.5%, it's worse, about 2% of our genome codes for proteins. Oh, why do we have all this extra junk? What's all this extra baggage we're carrying along? Well, it turns out that one person's junk may be another person's treasure. I like these new age websites tend to have some of the most beautiful graphics. So I stole this from some website that was talking about your inner genome and achieving quantum consciousness or something. But I like the picture. Look at all those yummy molecules. Who wouldn't want those? All right, so one of the most common constituents of junk DNA is endogenous retroviruses. So everybody's heard of HIV, the virus, the retrovirus that causes AIDS. Retroviral entities, it's hard to call them organisms. Some people say they're alive, some people don't. Retrovirus entities contain a little packet of genetic information and a needle to insert that information into cells. And then once that information gets into a target cell, it inserts itself into the genome of the target living cell, and the machinery of that cell begins copying the retrovirus DNA. Well, it turns out that every living thing on Earth, with exceptionally rare exceptions, where the editing and pruning and mechanisms have been particularly robust, nearly every organism has a DNA that is absolutely loaded with its history of encountering these endogenous retroviruses. So, rolling the cameras forward an arbitrarily long period of time, something like the HIV virus would, if it ran its course naturally, infecting huge numbers of humans without us trying to fight back against it with medicine, over a vast period of time, the survivors of that would have found a way, their genetic machinery, their cellular machinery would have found a way such that genes inserted into the DNA by that retrovirus were deactivated and made benign. It's that kind of benign information that constitutes a large part of our existing genome. It's a history of diseases written in our cells. We also have things called long non-coding RNAs. So one of the things that you may recall from the way genetics and the transfer of genetic information across generations happen, or even the expression of genes within a given cell, is that an enzyme comes along and splits the double helix a little bit, and then it transcribes the information in that double helix to something called messenger RNA. And then that messenger RNA leaves the nucleus with the template of that particular gene and goes out into the cell into something called ribosomes, where it's constructed into proteins. Well, there are these long stretches of DNA that are replicated into RNA, but the RNA that results from that is not messenger RNA. It doesn't leave the nucleus and get turned into proteins. And yet it makes RNA there in the nucleus. Isn't that interesting? Maybe that RNA that results from that is also junk. Maybe not. These long non-coding RNAs, the non-messenger RNAs, are generated by regions of junk DNA, but it turns out that many of them play a huge role in something called gene expression. They mediate the level of expression of other genes, or of genes, I should say. But they're really hard to find chemically. If you're working in a laboratory and you want to find these non-coding RNAs, the mechanisms that we currently have to deduce their structure, their existence, and their function is not at all mature. They tend to be not very gregarious molecules. It's hard to get them to interact with the dyes and the kinds of labeling and the kinds of markers that we put into cells to identify unique molecules. And interestingly enough, about 400 long non-coding RNAs are conserved across 300 million years of animal evolution. That seems to hint that maybe they're important. And if you have a feature of a cell that is conserved across species and across vast amounts of time, maybe they're not as junky as we thought. We also have junk that's in the form of insulation that says there's no gene here, move along, right? And other stuff that says, oh, hey, come over here, I've got a gene for you, right? So these kinds of signaling mechanisms are also encoded in DNA. And then we have copies of genes. Many times a given chromosome will have many different copies of a given gene, most of which are disabled. These are often called retrogenes, inert copies of genes that otherwise already exist. And then we have these things called telomeres. So what I'm going to do is I'm going to, one of the other interesting things is that these non-coding RNAs have an aspect that is intriguingly similar to something we know about proteins. It turns out that it's more than just the chemical properties of a protein that is relevant. Its shape is also highly relevant to bioprocessing. You may have heard of a project called Folding at Home. What that particular product does is it's a free piece of software you can install on your home computer, and it'll run while you're not otherwise acting on your computer, while your computer is idle. And this Folding at Home program looks at all of the amino acid bases, or all amino acids, it can't be an acid and base, amino acids in a protein. And using those, it tries to figure out how that protein folds in on itself and what the active regions are. Once it folds into this complex shape, there will be loops, there will be little charged bits that have a relative positive charge, there will be other little bits that might have a relative negative charge. And figuring out the folding mechanism of proteins and why that shape is active, bioactive, is a really hard thing to do. Well, it turns out a lot of these non-coding RNAs also form shapes, and it may be in many cases that the shape is what's really important. So this particular image shows two non-coding RNAs with very different chemical signatures but similar shapes. So there's more to look at than just the chemistry of a particular outcome. So when you look at a human and you look at our DNA, and by the way, this isn't limited to humans, every other organism has junky DNA as well. We've got all kinds of extra gunk in our genome. So what is that? Well, the first one is something called retrogenes. I talked about that. Ancient retroviruses that have almost in all cases been turned off. We have repeats. One of the things that we have is long interspersed genetic elements, liges, and short interspersed genetic elements, signs. There's a word for everything. What a language, huh? There's also non-protein coding sequences. We talked about that just a moment ago. Some are highly conserved across all species. We've talked about that some are conserved across at least 400 million years of evolution. Technically, everything that isn't part of our gene encoding data load in a DNA molecule is non-protein coding. So all of these other things that I'm talking about in this circle all make up the 98.5% of our genetic material that isn't gene encoding. Then we have the thing that everybody's familiar with, the protein coding sequences. That's the stuff that was previously thought to be the only important part. And then we have telomeres. So let me talk a minute about telomeres. So on the end of any given gene, in many cases, is this sequence of additional segments of information. Think of it, one of the ways I like to think of it is a rattle on a rattlesnake. You know how that's made up of little nodules, and the number of nodules can correlate to the age of the snake? Well, telomeres kind of have an inverse relationship like that. The number of nodules in a telomere kind of count down as a cell divides over time. Right? Many cells you have reproduce on the order of several days to several weeks. Every time a cell undergoes a duplication, and then the original cell is programmed to die off, one of the rattles gets knocked off. One of those telomeres gets knocked off. And over time, those telomeres essentially count down the number of times that that cell is able to reproduce. So one of the sets of investigations that are currently ongoing with respect to aging involves the role of telomeres play in the aging process. Telomeres are not a huge quantity of DNA in terms of the genome, but they're very important. Next is called enhancers. They're small molecular handles for transcription enzymes to anchor to while coding a gene. Now, here's a very interesting thing. These handles that say bind here because I've got a really good gene for you, they tend to be thousands and thousands of DNA rungs away from the gene that they're encoding. So one way to think about that is that the enzyme that reads a DNA segment may be shaped in such a way that it's got a piece over here that binds to the DNA where the enhancer is, and then it's got this long stretch of molecular machinery before it comes to the reader piece, and it has to span that distance. So in all cases, we find that the enhancers that say, I got a gene for you, are far away from the gene they actually encode, or the gene that is related to them. And then we have something very similar to that called promoters. And promoters say, the gene starts here, right? Very, very demanding little pieces of molecule. And then we have regions that are implicated in epigenetics. And we're going to talk a lot about that in a few minutes. Epigenetics, the short answer is it's two sets of chemical modifications that code for the amount of genetic expression of a particular gene. And then we have 3D interactions. And I talked about how those non-coding RNAs as well as proteins can fold in shapes. Well, it turns out that the DNA molecule itself, although we often imagine it as this twisted ladder, it has some very interesting properties three-dimensionally. It tends to be pulled and twisted and stressed in such a way that it acquires very interesting and complex 3D shapes. And some of the pieces of DNA itself are implicated in guiding the molecules that do the twisting and tugging and pulling that create these shapes. So even the shape of this double helix, you can imagine taking a bath towel, you know how you twist it up, and as you twist it and twist it, it starts making these strange knotting patterns. Well, DNA can do the same thing, and sometimes those knotting patterns have been co-opted by evolution to encode biorelevant information. One of the ideas that I've talked about in conversation with people is that evolution and biochemistry is kind of a kludge. You know, if something's laying around and it can be used or co-opted to be used for something or reused for something new, it will be. It's a mess. I don't know, nobody could have designed such a chaotic system. And then there's splicing, right? So it turns out there's a little bit of good news. Remember how I said you only have 20,000 genes? You actually make a lot more than 20,000 unique proteins. Well, the old standard dogma in biological circles regarding DNA and genes is that one gene coded for one messenger RNA that then made one protein. Well, it turns out that a given gene, if you cut it and splice it in different ways, well, why not reuse that same gene to make different proteins? Remember the kludge idea. Hey, look, we've got this perfectly good gene here. I bet if we change that, we could make something, instead of a skin cell, maybe we can make some transparent cells and co-opt those to be the lens of our eye, for example. So small changes, splicing changes in the transcription of a given gene can lead to many different messenger RNAs and many different eventual proteins through splicing. There's insulators, I talked about that a few moments ago, provides non-coding. Well, in order to get complex shapes, you've got to have material to work with. So it may be that some of these insulators don't just keep the transcription of DNA from exceeding its intended bounds or appropriate bounds, but the insulating DNA material also provides enough rope to hang yourself. Well, enough rope for the DNA folding to occur in such a way that the outcomes are biologically useful. So just providing runway, in a sense, is a useful function of DNA. And then there's a special part of a DNA molecule called a centromere, a special way that it binds itself to the spindles that it wraps tightly around. In one particular place, it forms something called a centromere. And this is what, whenever a cell divides and reproduces, this provides kind of a hitching post for the spindles within the, or for the transit lines within the cell to hook to these spindles and pull chromosomes apart, or to pull them into daughter cells. And there's even some recent thinking that the centromeres and the coding around this function, the non-DNA related coding of the backbone of the spindle that the DNA wraps around, is implicated in many diseases. Just like genetic errors are sometimes 100% correlated with particular disease outcomes, and sometimes they're more loosely or very loosely correlated with particular disease outcomes. This non-DNA part, the spindle part around which DNA wraps itself, has also been implicated in diseases. Well, if it's implicated in diseases, it's probably also relevant to normal functioning. So you've got this complex, three-dimensional, chemically diverse, highly spliced, multi-encoding system inside of every cell, and it's just a monster to sort out what's going on in there. The old idea of one gene for one protein, and that's the end of the story, is completely wrong. All right. I talked about chromosomes. All of your DNA, every DNA component for every creature on Earth is in chromosomes for every eukaryotic cell. Eukaryotic are cells with nuclei, and they tend to be the cells that are in complex multicellular creatures. We talked about eukaryotes versus bacteria and archaea, which are other whole kinds of cells, and generally just single-celled organisms. We talked about the distinction with some of those back in our first lecture. But it turns out that our DNA is packed into these chromosomes. Here's a picture of the 23 chromosomes in a human. If you get two X's, you're a female. If you get an X and a Y, you're a male. There are many diseases and pathologies associated with getting two X's and a Y, or other combinations of chromosomes, too many in a given chromosome rather than just a single pair. Let's talk a little bit about inheritance and the way that that works. We're going to talk about the fact that sex cells, oh, not this, this is an Armani ad. Different. different kind of sex cells. So everybody probably is familiar with the idea of the sperm and the egg, and it turns out that each of those, called gamete cells, only has a half of the complement required to make a fully living, viable cell. Right? There's only, instead of 23 pairs of chromosome in an egg and in a sperm, they only have 23 chromosomes, lacking a paired partner. And one of the most amazing things about the way biology works and sexual reproduction is a concept called crossing over that I just couldn't resist. So it's not this kind of crossing over. This guy thinks that you can talk to ghosts. It's a crossing over of once a sperm cell and an egg cell meet, or actually when they're being first generated by the parent cells, quite literally parent cells. You've got a copy of a chromosome from your father and you've got a copy from your mother, and each of those generally has the same sequence of genes on it. And I'm not going to go through dominant and recessive genes because I want to spend some time talking about epigenetics. But in general, you take your pairs of chromosomes in the parent cells, and there's this process where they cross over one another, and then they get randomly chosen to come from your mother's piece of the chromosomal DNA or your father's piece of the chromosomal DNA. And one of the questions I had is, well, what's the source of the randomness? Right? Is it really random, or is there some other process in the cell that says this gene for eye color is going to come from your father, and this gene for toe size is going to come from your mother? Is there something that specifies that in advance? And it turns out the best I can find is that the answer is no. It is literally random. And like many random processes in nature, it probably has a thermodynamic underpinning. You know how molecules tend to jiggle around if they're heated, and they bounce off in almost literally unpredictable trajectories if you have enough of them. That same kind of thermodynamic inspired randomness is what's implicated in randomly selecting genes and pieces of junk from either your paternal or maternal copy that you have within your own DNA that gets put in my case, into a sperm cell, or for a female, the same process happens. Her father or her mother's chromosome get randomly recombined to make a single chromosome. And then those go into offspring. And if you count up the ways that that a single person can combine the material on the DNA they get from their mother and their father, it's over 8 million. And if you count up the number of ways that two people in sexual reproduction can create a unique offspring, it's more, I believe it's more than the number of stars in the universe. It's one of the reasons that it's really rare to find two people that are identical unless they come from the same fertilized egg cell. It's not likely to happen. You can have children every 9 months for all of your reproductive life and have essentially zero chance that any two of them will be the same. All right. So here's an interesting question that people started asking soon after DNA was discovered. If nearly every cell in your body has the same copy of the same genetic instructions, and I say nearly every cell because things like red blood cells don't have a nucleus and don't have DNA, so I always have that caveat in there. But for all intents and purposes, of the 50 trillion or so cells in your body, they all have the same copy of the same genetic information. Then why is an earlobe cell different than a kidney cell? right? They've got the same set of instructions. They've got the same blueprints, if you will. Why are they different? Well, it turns out that genes can be turned off. And they have to be turned off. Whoops. I needed to turn my page, not my clicker. They have to be turned off, or every cell in your body will express every possible protein, and that just won't work. So, just the fact that a single fertilized egg cell can turn into a whole organism implies necessarily that there's some kind of control mechanism in a cell to regulate the expression of proteins. This is where the notion of epigenetics begins to become relevant to science. Epigenetics takes the idea that there are mechanisms for controlling gene expression a little bit further. One of the ideas that's common in current thinking is that the function of little pieces of RNA that cause different parts of genes to turn on and off, or different genes to turn on and off, the function of the RNA to control gene expression may have arisen as a way to keep lateral gene transfer and viral DNA in check, right? You don't want that HIV virus that gets into your cell nucleus. Really, that's not good for your species. You want to be able to turn that off. So, early in the history of life, having evolved some mechanism for turning genes off was likely quite critical to viability. Well, if you can turn a gene off, why not turn it back on again? Maybe you turn it on only in response to a particular stimuli. One of the things that's important in biology, one of the terms, a couple of the terms you'll come across, is phenotype versus genotype. Every cell has the same genotype. It's got the same blueprint. But the phenotype is the body that results, the organism that results. So how is it that the genotype is correlated to the phenotype? How does that work? What turns genes on and off? We can even look within a species. This is, I think, a Great Dane and a Chihuahua, and yet they're the same species. How can the same species exhibit morphologies or phenotypes that are so vastly different? And then we've got our froggies, the amphibians, right? Frogs go through a phase when they're tadpoles, and then when they're kind of this hybrid thing. I don't know, does that actually have a name? And then full-on frogs. How is it that genes control turning on different physical manifestations within the same organism? And as if that weren't bizarre enough, we've got this. So, not only are genes turned on and off during the development of an organism, when it's moving from being a single fertilized cell to being a viable offspring, but even during the lifetime of a given offspring, different genes can be turned on and off. And that's what happens when a caterpillar morphs into a butterfly, for example. Evidence shows that bits of RNA, and not the messenger RNA that transcribes gene information, but other kinds of RNA are implicated in gene control. These are tortoiseshell or calico cats. Now, in these cats, which are always female, because the instruction to say make some orange fur here only comes from a female. In calico or tortoiseshell cats, the orange hair cell follicles are completely and randomly activated. So you can find calico cats that are from the same litter that have vastly different coloration. And it turns out that when a patch turns on orange, it tends to affect the cells around it a little bit. And so you get not just individual hairs that are orange, but patches that are orange. These are a couple other cats. Now, I want to call both of these calico cats, but you'll notice that the cat on the right doesn't have any orange. These cats are named CC and Rainbow, and they are clones. So, the same nuclei was implanted into an otherwise fertilized egg that turned into this cat, and yet this cat randomly turned on 0 orange hair follicles. Right? Remember, the random thermodynamic processes at a molecular level can control a lot of this. Speaking of cats, you never have too many cats, right? These are descendants of Ernest Hemingway's cat. Do you notice anything interesting about them? Yeah, it's got an extra toe right here and right here. Now, this wasn't in the book I read, but something occurred to me about this. Doesn't this look kind of like a thumb?
Yeah.
Now, that's an epigenetic change. There's no gene that says, hey, make a thumb. It's an epigenetic characteristic that makes another digit appear. So maybe things as complex of whether or not, I'm not saying that humans have a thumb because of epigenetics, but it's an example of how a purely epigenetic phenomena that can be heritable across the generations can lead to noticeable morphological changes. One of the things, this is Alan Turing. He was his character, or a characterization of him, was in the Enigma Machine movie recently. He was a computer scientist, among other things. One of the things he came up with was a mathematical way for expressing reaction diffusion processes within living things to be responsible for things like the stripes on a zebra, or the stripes on a zebra fish. And it may be that the same kind of reaction diffusion chemical signaling across groups of cells is implicated in epigenetics, in turning genes on and off as well, in a patterned way. It's all very complex, and it means that there's more going on than just genes. So hopefully you don't feel so bad about having 85% of your DNA in common with a zebrafish. I'll take a little tangent for a minute and talk about stem cells. Remember the hubbub a decade or so ago regarding embryonic stem cells? The idea was that if you take a cell out of, once a fertilized egg begins to divide, but before it has differentiated into different tissue types, you take one of those cells and it is what's called pluripotent. It could become any potential cell type, any organ type, any tissue type. And there was a lot of ethical concerns about whether that was something we should do or not. I'm not going to address that specifically because it turns out that in 2012 a couple people won a Nobel Prize for having discovered that nearly any cell can be turned into a pluripotent stem cell without having to use embryonic cells. These are the two people who won that Nobel Prize. And you can essentially take a cell that has become a skin cell or a kidney cell or an earlobe cell and cause it to reset back to being essentially an unfertile or a fertilized egg white cell, with a couple of differences that I won't get into. One of the things that might allow is for eventual organ replacement, organs grown in a dish from your own cells. This is another famous clone. This is Dolly. One of the problems that is implicated in cloning is that oftentimes the messages that say turn this gene on and turn this gene off don't get stripped off when you're implanting that nucleus into an egg that is capable of dividing and forming a new organism. So sometimes you carry shortened telomeres with you, right? If the parent cell that was the source of the cloned nucleus had shortened telomeres, then the lifespan of the resulting creature may be abbreviated. So cloning tends to be fairly problematic, and some of the epigenetic effects can be relevant. But there are currently investigations underway to clone a mammoth, based on the fact that 99.4% of its DNA is identical to current Asian elephants. Recently there was a 40,000-year-old mammoth discovered in Siberia that had tissue and blood still relatively well preserved. That mammoth is named Buttercup. Another bit of a tangent, something called CRISPR and the Cas system. This will result in a Nobel Prize shortly. People were somewhat surprised that it didn't result in one this year. The idea is that using a genetic defense mechanism that is in simple cells, single cellular life, the prokaryotes we talked about before, we can co-opt that mechanism for maintaining and editing their own genomes and apply those to complex organisms to do exceptionally precisely controlled genetic editing in a living organism. It has great potential for curing diseases or doing genetically modified organisms that are more nutritious, more viable, etc. So this CRISPR system of genetic immunity will eventually result in a Nobel Prize. And there's all kinds of companies that are being formed to take advantage of this new technology. So let's talk about gene control. How does epigenetics work? Formally, epigenetics is defined as having exactly two chemical signatures. One is something called histones. Remember I talked about the spindles around which DNA coils? That's what these things are. These little green blobs are schematically representative of those histones. And these are alkaline protein spools around which DNA winds. Remember what the A in DNA stands for? Acid, right? So the spindles are alkaline. Alkalines and acids tend to want to be together. So these alkaline protein spools form the spindles around which always, in every case, 147 base pairs wrap. So one of the things that can happen from these is that the histones can acquire different sort of molecular decorations that allow them to ever and even more tightly bind the associated DNA strands so that that DNA is not at all accessible to enzymes that want to make RNA. It locks it up, and it can lock it up for the life of a cell. There's another mechanism called methylation, where another different kind of chemical decoration is put onto DNA that says, hello, I'm here, I'm free, play with me, right? And that kind of decoration can either enhance the expression of an associated gene or repress it, depending upon whether these little spindly arms grab each other and make a tighter knot or not. About 3% of the human gene tends to be methylated at any given time. Remember, only 1.5% codes for genes. So we have twice as much methylated DNA with these little handles and signals on it as we do gene encoding pieces of DNA. These things can be relatively easily turned on and off. These tight spindles, the histone spindles that lock DNA away, are really hard to get rid of, and in fact most cells, once they develop tightly coiled histones, it can't be undone. This is another view of how that methylation may happen. It's a kind of ornament in the DNA structure. So what are some of the effects of these kinds of ornamentations of cells? Well, Audrey Hepburn, famously beautiful actress, was very slender, and her phenotype, the fact that she was slender, is likely a result of something called the Dutch hunger winter. So she was a teenager in Holland during the Nazi occupation. And during 1944 and 45, the ports were blockaded and a lot of people were starving. And it turns out that those kinds of starvation effects that happened early in her childhood likely resulted in low body weight for the rest of her life, regardless of how much she ate. And it's not just her, it's other children that were developing their metabolism and their bodies during this time that see the similar effect. Another study called from Scandinavia called the Epigenetics study found that nutritional effects showed evidence of impacting great-grandsons of great-grandfathers that had particular eating patterns and nutritional availability. It turns out if you had a lot of available nutritious food, your grandsons and great-grandsons would be more likely to have obesity-related problems like diabetes and have shorter lifespans. If you, as a great-grandfather, were in a period of famine and stress, then it's more likely that your offspring and your next offspring would have better abilities to maintain body weight and to have a healthy metabolism. So you'd have to go through a lot of pain to make sure that your great-grandchildren had a successful life. Something many of you are familiar with. Recently, there was a study that said that scientists can predict with 70% certainty whether a given individual is homosexual or not by looking at the epigenome, looking at the way different genes are expressed or not. 70% is very statistically relevant. Another study showed that the body mass index was raised for sons of men who began smoking early in their own lives. So these cross-generational effects of turning genes on and off seem to be, in some sense, transmitted across generations by virtue of the germ cells or sex cells. Typically, when a sex cell is originated, a lot of these decorations are stripped off, you'd think. And in many cases that's true. But in some cases, some of the decorations of the parents are heritable. Same kinds of effects have been seen for cortisol, stress level, lots of nutrition, and environmental chemical related effects. Depression, stressors associated with depression might lead to epigenetic changes that are somewhat heritable. Running a little short on time, I'm going to breeze through this. This shows the amount of inherited information from a great-grandfather through time. I won't go through the math unless somebody would like to talk about it afterwards. So we've got these great factories in our bodies, our metabolic factories, the cellular factories that make proteins and express proteins and whatnot. And they're constantly adding on these decorations to our DNA, and as a result of the food we eat, the vitamins we get, etc. Especially early in life. For example, too little folate or choline before or just after birth leads to undermethylation for life. The ability to regulate gene expression is impaired for life if you lack certain nutrients as a developing child. Perhaps even if the mother lacks some of those nutrients. A lot of these are brand new, relatively new within the past 5 years, studies, and the data is so complex that the correlations have been seen, but the underlying specific causes are really hard to pinpoint, especially when you've got something like RNA that folds into complex shapes and isn't easy to detect in the laboratory. So a lot of these are inferred. This kind of environmental dependence of the epigenome on what we eat or ingest leads to problems like this with things that we inhale or inject. Cocaine, opioids, alcohol all lead to changes in gene and DNA region methylation, or the epigenetic expression level of different genes. Genes that often are implicated in brain function. The brain has some of the most complex epigenetic expression machinery in the human body. So if you're going to be messing with epigenetics, if you're going to decorate and undecorate cells, neurons tend to be highly sensitive to those kinds of processes. And as we learned, when you introduce tight coiling around histones, that tight coiling, that histone suppression of a gene, once it is acquired, perhaps through some addictive process, it stays for life. Which lends credence to the idea that addiction can't be cured, it can only be managed. Environmental factors such as stress seem to turn on epigenetic responses that increases someone's susceptibility to addictive behavior. So there's these giant feedback loops in biochemistry and the things we eat and the environments we find ourselves in. Here's a couple of articles about epigenetic inheritance being relevant across generations. I go on to the next one. Look at the date.
Wait till the first.
Right. Right. Right, so there's this argument in biology about nature versus nurture. Well, it turns out it's not an either or. It's a complex and literally chaotic interplay between them that is relevant our whole lives and in the lives of our parents and perhaps our grandparents. We don't fight against nature so much as we integrate into it, and the environment in turn affects us in profound, entangled, and multiform ways. There's a great set of feedback loops inside each living thing, including us. Nature and nurture integrate and compound in astonishingly complex ways. Epigenetics has again shown us that science uncovers truths about nature that are truly astonishing and unexpected. Our simple either-or view of nature or nature, nurture or nature, is shown to be embarrassingly inadequate. We've found this rabbit hole of complexity when we've looked more closely at biological processes and in this particular case, in the processes implicated in genetics and gene expression. It may seem endless and infinitely regressive, but DNA and the RNA components and the epigenetic chemicals constitute a finite set, almost innumerable, but still finite. So in principle, this kind of mechanism can be understood, and it is amenable to scientific investigation. In practice, there are trillions of billions of ways that molecules can fold, decorate, splice, and react. And from a practical standpoint, it is unlikely that we will ever get to the bottom of it. But my goodness, it's fun trying. And hey, that's life. Thank you very much. Any questions? These are some of the books that were inputs into my brain for this talk.
Can you expand on the 70% business about homosexuality, predictability?
Yeah, so article says that by looking at different histone levels and methylization of particular genetic regions, that these researchers could predict with a 70% confidence whether a given person is homosexual or not. One of the first epigenetic phenomena I remember reading about, I happen to be gay, and so this topic interested me well before I had any experience with genetics. I remember reading that during the London Blitz, mothers who were pregnant during the Blitz and underwent tremendous stress, like psychological, emotional, and in many cases physical stress, were more likely to give birth to homosexual offspring. And so that kind of environmental impact on sexual preference is not surprising, given that environmental impacts affect so many other complex aspects of being human. And then I assume, building on that research as well as subsequent research, these scientists have said that they can predict with about 70% certainty, given epigenetic signals, whether or not a given individual is homosexual. Other questions? Yeah.
You mentioned that these new developments could maybe discover new disease, ways of dealing with disease. I read last spring that a cure for AIDS had been developed, and rather than getting into the details of it, essentially the AIDS virus has a case over it, so it can't be attacked. And it attaches to a CD4 cell, which is a blood cell, but it attaches only to those CD4 cells that have an expression of a protein called CCR5, okay? If it doesn't have C, but that's where it latches.
It's a unique signal that says, hey, I'm an HIV.
Well, no, it actually attaches like a space station.
Okay.
And it then goes into the CD4. It slices up the CD4, it makes itself. But they've discovered a way of taking a person's CD4 cell count, removing that protein, or that whatever it is, that makes the CCR4 expression.
Right.
So that ever after that, the white blood cells of that person
Are ignored by it.
are ignored, and so it can't reproduce itself, and they're cured.
Wow.
They wondered why why sex workers in Central Africa, some of them never got AIDS, and it was because they didn't have that CC.
That particular mutation.
That expression on their CD4 cell count. But somehow by taking that out of the person's CD4 cell development, they're cured.
Amazing.
That's exactly what you were saying. That these are going to lead to amazing cures.
Yeah, thank you for sharing that. Other questions? Yes, ma'am.
How do they measure these astronomical numbers of infinitesimal things?
Oh my goodness. So there's mechanisms. Once you have a piece of DNA, there was a Nobel Prize, I think it was maybe in the 80s, awarded for, given a small snippet of DNA, there's a chemical process that can reproduce that ad infinitum, so you end up with a macroscopic sample of identical DNA. And then once you have that macroscopic sample, you can start measuring its properties instead of having to deal with one particular DNA strand. So there are different mechanisms like that where they can essentially amplify the amount of material that they're measuring, and that obviously amplifies any signal associated with the property of that material. Another way is marking. They can use radioactive or quite literally fluorescent molecular markers. They can pin them into some of this biological material or DNA or into a cell, and then literally watch these fluorescent markers as the cell divides or carries out its metabolism. So there's all kinds of ingenious ways about tagging and tracking and amplifying a lot of these signals that are very subtle. Any questions? Yes, sir.
over time, has there been any kind of a characteristic that was initially associated with the epigenetic phenomena or with mitochondria, your DNA, or with the DNA in the nucleus, shift from one association to the other?
Yes. Yeah, so I don't
Repeat the
know.
question?
Oh, yeah, the idea is over time, has there been cases where DNA that was expressed in mitochondria might shift, say, into the nucleus, or where something that is an epigenetic phenomena may instead be encoded directly as a gene. So the last part of that question, an epigenetic phenomena turning into a genetic phenomena, I don't know of any examples of that. I didn't come across any of those. I'm not a practitioner in this field, by the way, so don't take my word as gospel, but I didn't encounter any of that. However, there are certainly strong pieces of evidence to support the idea that the DNA in mitochondria, remember it's only, what was it, 247, I forget the number, a relatively small number of genes in mitochondria. And it's not enough to make the mitochondria a viable entity in of itself without the surrounding environment of a cell. And there's good evidence that the genes have migrated out of mitochondria DNA into cellular DNA. It's offloaded some of its metabolic and regulatory processes into the nucleus. And the DNA within the mitochondria itself, it's believed that that's the minimum amount of DNA possible in order for the mitochondria to continue to do its job as a metabolic battery. That's, that discussion is given much more thorough treatment in the book that I used as reference to the possible origin of life on Earth, which was, I can't remember the name of the book. I've read so many lately. But I refer to it on the video for the origin of life talk. And Nick Lane is the name of the author. And it talks about mitochondria and their evolution. Yeah.
All these books that you read for this, are you buying these books and have a great science library? Are you putting them on device, or how do you get your information?
So last year, the American Humanist Society provided a grant to our chapter for supporting this science lecture series, and I used some of that money to buy these books. And typically I buy digital books, they're on my Kindle. My physical library is so large that it is overflowing its shelves. So I've transitioned to electronic readers. But yes, I buy all these books. I don't tend to check out or rent books. Yeah.
If I understood this right, sometimes there's epigenetic decoration in the genetics of a cell that is kind of temporary or it's limited generationally, but then there's, you said, histones that are so tightly wound that they permanently remove DNA from consideration.
Right. Yeah, so there are two types of epigenetics has been formally defined as the effects of these two chemical processes. Methylation and histone entrapment of portions of DNA. Excuse me. Methylation is undoable and changeable. That's those little spindles that we're waving around saying, hey, play with me, or they bind each other and say, don't come here, right? And it looks like biochemically that's quite changeable. But once you've coiled up a piece of DNA very tightly around a histone, there are a few cases where that can be undone. So in one of the books, I think it's the Epigenetics Revolution, one of the things the author, she's the author of two of these books, by the way, Nessa. She says that in neurons, right, neurons tend to last your entire life. Right? The neurons you're born with are the neurons you have throughout your life. And you don't want them to act like kidney cells. So the bottling up of different genes in your neurons must be permanent. And that entails this kind of histone entrapment. There are times, however, when a given cell can and has shown that the histone entrapment can be undone. And often that happens when you're making sperm or egg cells. So all of that decoration that's been added to your cells over time must be removed in order to make a viable egg or sperm cell that's got essentially a blank genetic slate. But that process isn't perfect, and that's one of the reasons we see epigenetic inheritance occurring. Other questions? Yeah.
Some of the maybe undesirable or neutral genetic instruction, say for example some of the viruses, if somehow it was by accident or scientific design, someone was removed to make the human or animal genetics simpler, more straightforward, and without bad effects, would that impair the ability of that organism to produce fertile offspring with other encoded species that did not have that?
It depends on the change. So the question is, would genetically manipulating an organism affect its ability to have offspring?
Yeah, fertile offspring, yeah.
Yeah. Yes and no. It depends on what the change is and whether it's heritable or not, whether it manifests itself in the germ or sex cells that organism produces. So yes or no. Okay.
Another interesting aspect is we're just beginning to explore is the whole idea of orphan genes, where portions of the junk become separated from and become active genetic material,
Right.
producing stuff. And it just sort of wanders around. And again, we're going back into, is this inherited or is it inheritable?
Right.
And at this point, there's an awful lot of just question marks. But again, the process is junk becomes active, and then what? Does this lead into the whole process of evolution, and if so, how? And, you know,
Absolutely. It's amazing and complex and mind-blowing. The idea that some pieces of junk DNA can actually start coding for proteins, right? They can become activated through some mutation. And maybe that's a mechanism for developing new traits over time. Now, in most cases, statistically, those will be detrimental, but occasionally a beneficial trait may develop. All right, thank you very much. Next time we're going to talk about Gaia, the way Earth systems, physical systems, and life feed back on one another. And we'll be going to lunch imminently at a nearby restaurant. If you want to know more, come up and see me. Or otherwise, thank you and have a good day.
that takes care of it. That's fine.
Wow.